Hello everyone, this is lecture 5 and in this lecture we'll learn about bisubstrate reaction. In lecture 4 we have seen how the substrate concentration affects the rate of a simple enzymatic reaction with only one substrate molecule. That means we have we have learned about the effect of substrate concentration for monosubstrate reactions. However, in most enzymatic reactions, two and sometimes even more different substrate molecules bind to the enzyme and participate in the reaction. Okay, so for example, the reaction which is catalyzed by an enzyme by the enzyme hexokinase, ATP and glucose are the substrate molecules, and ADP and glucose 6 phosphate are the products. Okay, so in this lecture, we'll mainly talk about the reactions that require two substrates and yield two products. These kind of reactions are also called by reactions. Okay, and please keep it in your mind that there are reactions where more than two substrates, okay, can be involved and more than two products can be produced. Okay, so however, we can name this kind of reaction based on the number of reactants and the products. For example, if a reaction there is one substrate and three products, that reaction is named as unitar reaction. Okay, uni means one and tar means three. So in the same way, if in a reaction there is one substrate and two products, it will be named uni bireaction. Okay, so according to this, in case of bisubstrate reaction, there are two substrates and two products. So that's why we call them bi reactions. Okay, and 60% of known biochemical reactions, they are actually bisubstrate reactions or bi reactions, and almost all of these bisubstrate reactions are either transfer reactions in which the enzyme catalyzes the transfer of a specific functional group from one of the substrate to other. Okay, so that means most of them, I mean more than 50% of biochemical reactions are bisubstrate reactions, and in this kind of reactions, mainly transfer of functional group occurs. Okay, uh, these are two examples of bisubstrate reactions. So, for example, if you consider this one, this reaction it occurs between ethanol and NAD catalyzes by alcohol dehydrogenase. So if you look at the product, you can see that in this reaction, one hydrogen ion was transferred from one substrate to another. Okay, and actually in most of the bisubstrate reactions, transfer is the main thing that happens. Okay, and bisubstrate reactions are classified into two groups, into two types: sequential reactions and double displacement reactions. The double displacement reactions are also called ping pong reactions. Okay, so first we'll learn about the sequential reactions. Okay, so in case of sequential reactions, first both the substrate have to bind with the enzyme, and the complex formed is called ternary complex. Ternary complex. That means when both of the substrates are bound with the enzyme, this complex is called ternary complex. So in all the sequential reactions, first the ternary complex have to be formed. Okay, then product will be formed. Okay, and anyway, so in such reactions, I mean in the sequential reactions, the group being transferred, for example X, is directly passed from substrate A to substrate B, okay, which yield product P and Q. Okay, so as one functional group is displaced from one substrate and transferred to another substrate, sometimes these reactions are also called single displacement reactions. Okay, and sequential reactions we can also further classify them. Okay, so sequential reactions, they are classified into two groups. One is called ordered mechanism and another is called random mechanism. So, and this name arises depending on the binding of the substrates with the enzyme. In case of ordered mechanism, the substrate have to bind with the enzyme, maintaining an order, such as, for example, first substrate A will bind with the enzyme, then substrate B will bind with the enzyme, then product P will released, then product Q will released. Okay, this will follow a ordered fashion. But in case of random mechanism, either substrate can bind with the enzyme first. Suppose, okay, and there is no order which product will be released first. Okay. However, in either case, it can be ordered or random. Ternary formation of ternary complex is must. That means first both the substrate have to bind with the enzyme first. Then the reaction will proceed. Okay. And in the ordered mechanism, the binding of first substrate is apparently required for the enzyme to form the binding site for the second substrate. Okay, so that's why actually in ordered mechanism, a ordered is essential, because if the first substrate don't bind with the enzyme, the binding of the second substrate will not be will not occur. Okay, so that's why ordered is necessary, essential here, because when the first substrate bind in the enzyme, it's influence the enzyme in a way that then the binding of the second substrate get activated. So that's why the second substrate can bind them. Okay. In contrast, in case of random mechanism, no such things happens. Okay, both the binding sites are present there in the active form. Okay, that means both the binding sites are present on the free enzyme. So we can represent this notation of sequential reactions by this graphical way, which was developed by a scientist, W. Cleland. Okay, and in his representation, represented he designated A for the first A and B for the substrate, okay, and P and Q to for the products, okay, and E represent the enzyme. So if you see this, first the A bind with the enzyme and form the EA, then the binding site of B is activated, so B now can bind with this complex and EAB is formed. This is called a ternary complex. Okay, and intermediate products are formed like EAB, EPQ, and then one product P is released. Finally, product Q is released. So everything happening in a orderly fashion. Okay. Okay, so this is an example of sequential ordered reaction. In this example, the enzyme lactate dehydrogenase, it catalyzes these reactions, in the reactions between pyruvate and NADH. Okay, in this reaction, one hydrogen ion is transferred from NADH to pyruvate. So, like, okay, and the product lactate is formed. Lactate and NAD is the are the products. Okay. So if we want to represent this reaction by this model, which was developed by W. Cleland, it will look like this. Okay. So in this reaction, this one. Okay, I forgot to mention you that NADH is uh, act as a coenzyme in this reaction. So first, the this coenzyme NADH will bind with the enzyme. Okay, then another second substrate pyruvate will bind with the enzyme. Okay, so this have to be done in orderly fashion. First the NADH will bind with the enzyme and then it will activate the binding site of the pyruvate, so pyruvate can now bind with the enzyme. Then first the product lactate will be released from this from the enzyme substrate complex, that is from the ternary complex. Then finally the NAD will be released. Okay. So uh, it was, I mean, this was about the sequential ordered reaction. Now let's move back to sequential random reaction. So in case of sequential random reactions, as I told you earlier, there is no order. Okay, I mean, any substrate can bind fast with the enzyme and any product can be released. Okay, such as if you uh, see this representation, say this is the enzyme. Okay, it can lead, this reaction can proceed in this direction. I mean, first A can bind, then B can bind, or in this direction, first B can bind, then A can bind. Then the ternary complex is formed, reaction intermediates are formed, and then from this complex either P can be released first, then Q, or either Q can be released first, then P. There is no order. Okay. This is an example of sequential random reactions, and in this reaction, it occurs between creatine and ATP, and catalysis, and this reaction is catalyzed by creatine kinase, this enzyme, and the products are phosphocreatine and ADP. That means one phosphor group is transferred from ATP to creatine. Okay. So now if we want to represent this reaction in this model, it will look like this. Okay. So I mean, either substrate, either ATP or creatine, they can bind fast with the enzyme. Okay. And then either phosphocreatinine can be is released first or ADP is released first. Okay, there is no orderly fashion. Okay. Now, I mean, we're done with the sequential reaction, and we also talked about the sequential ordered reaction and also sequential random reaction. Now we'll learn about double displacement, that is ping pong reactions. So in this kind of reactions, formation of ternary complex is not necessary. That means both the substrates don't bind with the enzyme first. Okay, so it can happen. I mean, this kind of reactions usually happen in this way. First, one substrate bind with the enzyme, then one product is released, then the second substrate binds, and the second product is released. Okay, so this is one example of this is one double displacement by substrate reactions. It all happens between aspartate and alpha glutarate, and this reaction is catalyzed by this enzyme aspartate aminotransferase, and the products are glutamate and oxaloacetate. Okay, and however, one defining feature of double displacement reactions is that an temporarily modified enzyme intermediate is formed in this kind of reaction. Okay, a modified enzyme structure is formed. Enzyme intermediate is formed in this kind of reactions. Okay, so if you look at this example again, so let's see. I mean, let's try to understand. I mean, what we mean by this one, formation of enzyme intermediate. So, I mean, when this reaction occurs, it occurs in this way. First, the aspartate, I mean, aspartate and alpha ketoglutarate are the substrate for this reaction. So first, the aspartate bind with the enzyme, then the NH3 group of the enzyme, it transferred into the the NH3 group of the aspartate is transferred into the enzyme. I mean, this NH3 group, is transferred from aspartate to the enzyme, and this enzyme NH3. Okay, this temporary modified enzyme intermediate is formed, and during that time, oxaloacetate gets released. Okay, oxaloacetate is a product. Okay, I mean, when NH3 group is transferred from the aspartate, it becomes oxaloacetate. Okay, so oxaloacetate is released, and ENH3, that is a modified enzyme intermediate, is formed. Then the second substrate comes and bind with this intermediate enzyme structure. Okay, and then finally this NH3 group is transferred from the modified intermediate enzyme to alpha ketoglutarate. Okay, and the alpha ketoglutarate becomes glutamate, and enzymes get back to its original form. Okay, so that's why it is called double displacement. Look, I mean, this amino group was transferred. I mean, why these reactions are called double displacement or ping pong? Because this amino group was transferred in this reaction. I mean, amino group was transferred from aspartate to glutamate, but this transfer was not a single step reaction, right? First, this amino group was transferred from aspartate to the enzyme, then again it was retransferred from the enzyme to glutamate. So double displacement occurs here in a ping pong fashion. So that's why it is called a double displacement or ping pong reactions. Okay. So this is another example of a double displacement or ping pong reaction. So this in this example, the a phosphorus group is transferred from ATP to ADP. Okay, and nucleoside becomes, I mean, the NDP and or DNDP, it becomes NTP or DNTP. Okay, so why it is a double displacement reactions? Actually, who are the products? I mean, who are the substrates in this reaction? Adenosine and nucleoside. Okay, dinucleoside. Okay, so if we look this overall reaction, a phosphorus group was transferred from adenosine to the here, right? To nucleoside. Okay, here to here. But this was not done in a single step. First, the phosphorus was transferred from ATP to the enzyme. Okay, and this phosphorus, I mean, during this intermediate structure, the phosphorus was bind with the histidine group of the enzyme. Then finally, again, this phosphorus was transferred into this substrate and form the product. Okay, so ping pong. Okay, double displacement. That's why it is called double displacement or ping pong reactions. So okay, so anyway, that was all for this lecture. Thank you for your patience.